Hi, my name is Paul Walker. I work for Cyprotex, which is a ADME tox CRO, and we're part of the Evotec family of CROs. And today I'd just like to introduce you to some of our in vitro approaches that we're using in safety assessment. So as a brief introduction to the presentation today, so first of all, I'd like to walk you through one of the technologies that we use, which is high content imaging. And then also our application of high content imaging in 2D and 3D cellular models. Then I'd like to move on to the development of a cell stress panel, which we are using as part of a next generation risk assessment approach. And then I'd like to go through some examples from the high content imaging data that we've that we can show for you, showing a variety of images and biomarker responses. And then finally, I'd like to go through a recent publication that is currently in press, which is a collaboration with Unilever, where we've tested this new cell stress panel. So first of all, high content imaging. So high content imaging is quite a well-established approach across using in vitro models. There's lots of different high content imaging platforms available. Ideally, we like to do live cell imaging, and they are quite modular systems, so you can add things like confocal imaging. Most of them have automated platforms, so you can quite easily image in an automated fashion different wells of a plate, capture lots of different images, and then ask specific questions as to the health or responses within those cells. And we do that by adding a range of different antibodies or dyes. We identify individual cells, we identify things like individual organelles within those cells, and we can split those cells into nuclear regions or perinuclear regions or the cytoplasmic region, and ask specific questions on reagents that we use to dye specifically those cells. So we can add things like antibodies or dyes to look at specific cellular biomarkers and ask additional questions like location of those cells, location of those biomarkers, things like translocation to the nucleus, or changes in intensity across the cytoplasmic region, or indeed changes in kind of structure or mass of those particular dyes and antibodies. The nice thing about the reagents, where possible, we can multiplex those into a single assay plate or a single assay design, taking advantage of the different fluorescent properties of the dyes or differing secondary antibodies that can be used. So we also use high content imaging to look at 3D models. So here on the left, you can see we can derive different organ models by using either a hanging drop system or a ultra low adhesion plate. And the critical thing about developing and certainly imaging 3D models is the position of those tissues within the well. The nice thing about the ultra low adhesion plates is that they are round bottoms, so that means that we can, with a few different imaging techniques and processes, we can manage to get the microtissues in a single field of view at any particular well. So that means instead of having to hunt around those wells and find the microtissue, we're pretty confident that within a single field of view in the center of the well, we can find our microtissue, which basically just stops us getting things like edge effects and speeds up the image acquisition process where we don't need to hunt for those microtissues. And then that allows us to confocally image through these microtissues and determine, as I mentioned before, different biomarkers using a range of different dyes and antibodies. Here is just some data where we've used one of these 3D models, or well, two different liver 3D models. One where we've developed human liver microtissue, which is primary human hepatocytes, which we combine with Kupffer cells, and a second one where we just use the HepRG cell line, which we can form into a 3D structure. Both of these models develop quite intricate structures in a 3D model, so things like bile caniculi formation, they have good CYP expression. And the nice thing about them is the longevity. We get good CYP expression and they last out to at least 21 days in culture. What that's allowed us to do is test a reference set of compounds. So here you can see on the left a panel of 55 reference compounds. Basically, on the left column in red are most DILI compounds, and then we have in yellow the less DILI compounds, and then green the safe compounds. And what we've done is repeat dosed these microtissues for 14 days, and then using high content imaging, we've looked at a range of biomarkers. So here we've looked at a nuclear stain. We can also look at mitochondrial mass and mitochondrial potential with a dye, a specific dye. We can look at ROS formation, GSH content, and all of those different live cell dyes are combined in a single well. So we can determine these features from every microtissue, from every well. And we also determine cellular ATP as well as part of this set of endpoints. And we've looked at the predictivity of these different microtissues when we normalize the data to total plasma Cmax in humans with a cutoff of 25 times Cmax. And what you can see with this approach, we actually have achieved 100% specificity in both models and quite comparably high sensitivity in both the human liver microtissues and in the HepRG spheroids, with both models giving over a total of 90% accuracy. This data has been is in press and will hopefully be available soon. So next, I'd like to go on to the development of a cellular stress panel that we've developed in collaboration with Unilever as part of a tiered next generation risk assessment approach. As you've already heard, NGRA approaches from previous and strategies from previous speakers. So the first thing we set about doing is looking into a whole range of AOPs and different pathways and mechanisms that can lead to potential safety liabilities or potential risk of harm. And we identified a range of different pathways. So you can see we came up with 36 different biomarkers, again, which could be multiplexed where possible into specific assay plate designs. And we identified pathways such as DNA damage, inflammation, ER stress, metal stress, heat shock proteins, and things like that. So you can see it's quite a broad kind of capture-all type, well, not maybe capture-all, but certainly a comprehensive list of pathways to potentially look for safety liabilities. And we used initially the HepG2 cell model and looked at 3 different time points. You can see 1, 6, and 24 hours length of exposures where we're running HepG2 cells, where we looked at all these 36 biomarkers. So this is the simple illustration of all the different biomarkers, not necessarily the different how they are multiplex, but a list of those biomarkers under each of the mechanisms. The majority of them are established through high content imaging. So in blue are the high content imaging endpoints. Now, obviously, as I've said, that could be a range of dyes and antibodies to be multiplexed where possible. In addition to those, we looked at a range of cytokines, which we've determined with ELISA. In addition to that, we also have used the Seahorse platform, which measures mitochondrial effects and cellular mitochondrial bioactivity. And we can determine things like OCAR and reserve capacity, as well as ECAR, to determine mitochondrial effects and the health of the mitochondria. In addition to that, just as we did the 3D model, we can use Promega kits to determine ATP and LDH release. So just moving on to some of the data, here you can see with an exposure to cadmium across we typically run an 8-point concentration curve in triplicate. You can see with cadmium at 4 micromolar, you can see accumulation of metallothionein expression within those cells, which is seen with the fluorescence green. Typically what you'd expect is the transcription factor MTF1, which would get activated, leading to an increase in metallothionein expression, which would then sequester or act to sequester any free metal ion within the cell. Here looking at ER stress, again, a panel really of endpoints to look at potential ER stress. Here we've exposed the cells to tunicamycin at 10 micromolar, and you can hopefully see in those images various colors representing the different markers of ER stress. So we've used ER tracker to look at endoplasmic reticulum or ER disruption. BIP as a ER chaperone and a key marker for ER stress, and then finally CHOP as a specific endpoint for ER stress. Then mitochondrial, mitochondrial effects or mitochondrial oxidative stress. Again, you can see a range of images where we've dosed the cells to rotenone, which is complex 1 inhibitor of the ETC, and we can see mitochondrial ROS, specific mitochondrial ROS increases in response to rotenone, followed by PGC1 alpha activation, which again is a key marker in oxidative stress. And this was also linked to anti-immune inflammatory response by the expression of TNF alpha induced protein 3. So as part of this approach with Unilever, once we've established the cell stress panel, obviously we miniaturized that approach as much as we could to get the best assay design and as many biomarkers within a single plate type as we could, multiplexing up to 7 different endpoints. We obviously selected the cell type, an initial cell type to investigate, and looked at 3 different time points. We then selected 13 benchmark substances with known safety effects, and we've categorized those as either low risk or high risk. And then as we moved on then to kind of establish the dose of which we would expose the cells to, and we use this by looking at kind of clinical data for these chemicals and compounds, and PBK modeling to establish Cmax concentration. And this work is also in press and should be available soon. This is just a snapshot of some of that data. So we evaluated these 13 benchmark chemicals and drugs. As I said, they were characterized into low and high risk, as you can see on the left. And what we did was we determined the point of departure, so i.e. the earliest concentration of which we see a response in these 36 biomarkers, and then at the 3 different time points. And we've plotted that data against the Cmax, which you can see the dashed green line. And for most of the low risk compounds, you can see we see a clear separation, i.e. a much weaker response, if any response, to the 36 biomarkers, well below what the plasma Cmax concentration would be, except for the only compound that's close to Cmax or just above Cmax was triclosan, which is known to cause some mitochondrial effects, and the responses that we observed were specifically related to the mitochondria. With regards to the high-risk compounds, again, we pretty much saw the opposite, where many of the pods that were calculated from the 36 biomarkers were in excess of the 1x Cmax prediction of exposure, indicating again high risk. The only compound that we didn't pick up with such high threshold was diclofenac. Diclofenac may have not been picked up due to the low metabolic activity of HepG2s. Interestingly, in the 3D data that I showed you, even though you couldn't quite see the detail, diclofenac is picked up in a 3D long-term repeat dosing model. And as I said, this publication is pretty much close to being in press. So just to finish off the presentation, so introduced you to the development of a cell stress panel, which we think could form one of the NAMs that could be used in a tiered approach for next generation risk assessment. Panels made up of 36 different biomarkers. The initial validation obviously is within HepG2s, but many of these biomarkers could be used in a whole range of different cellular models and obviously assessed at different time points. And some initial validation of which we've used 13 different benchmark substances with the establishment of the point of departure and how we've been using that to normalize against Cmax as an IVIVE approach in risk assessment, and as I said, as part of a NGRA tool. Thank you.